Some examples.
Okay. Well, thanks, Hezi. And thanks also to Clinam for the opportunity to talk. I see things flashing up here that says my time's already over. And by the way, I'm not supposed to speak until 9:55 if you look at... So, but then the break is at 10, and I have to leave time for questions.
We do it in the Israeli way, forget about it.
Okay. So Hezi asked me to discuss how we measure size specifically, but also tailor this to show you how physicochemical parameters greatly influence biocompatibility. I'm using the wrong one. There we go. So for those of you that may have missed our Monday talk, just very briefly, NCL is a free resource that's available to anyone that has a nanotech application for cancer therapy or diagnostics. We're a formal collaboration among NCI, the National Cancer Institute, NIST, and FDA. When we receive particles, we characterize them under a three-phase assay cascade. The first being physicochemical characterization, then in vitro, and finally in vivo. And we generate data in support of a sponsor's IND filing, and specifically data that contributes to the CMC portion and also to farm tox. Over the last 10 years, we've characterized more than 300 particles, and 7 of those are now in clinical trials. So we have very good historical and empirical knowledge on the strengths and weaknesses of many of the particle types. Under physicochemical characterization, as I said on Monday, we have a portfolio of instrumentation that allows us to go after the questions that we're interested in, such as size and, I should say, orthogonal techniques to look at size as measured by DLS or EM or atomic force microscopy. Because I'll make the case through my talk that depending on which instrumentation you use, it's a different measurand, and you'll certainly get a different number. So you have to really understand the nuances of that and which parameter you're truly analyzing. So I'll start with something that's now fairly old and been published a number of times, and that is our observation and trends on how physicochemical parameters influence biocompatibility. So out of the very long list of parameters that we initially characterized when we started the NCL 10 years ago, we were able to narrow that down to 3 main influential parameters, being zeta potential, or more or less the surface charge, the size of the particle as monitored by the rigid core, and I'll define that here in just a second, and the hydrophobicity. And more or less that latter point is the degree of pegylation on the surface and its ability to avoid opsonization by plasma proteins. Here's what we see over the years. If you start at the upper left-hand corner, particles that are highly cationic or positively charged in excess of about 20 or 30 millivolts, positive millivolts, are cytotoxic. We see that both in vitro and in vivo. In vitro, they can literally punch holes in the membrane of cells, and under in vivo conditions, they will induce complement. So we consider those quite toxic. Under size is monitored by the rigid core, meaning the inflexible portion of the nanoparticle. We find that particles less than about 10 nanometers, and actually it's more like 8 nanometers, are cleared very quickly by the kidneys. We have some evidence that particles less than about 30 nanometers can be excreted by the bile duct. Coming up to the upper right, particles that are hydrophobic, meaning naked particles or inorganic, are very rapidly opsonized and phagocytized by the RES system. The body is very efficient at recognizing foreign material, and a nanoparticle, unless it's stealthy, is a foreign material. Then the sweet spot, as you've heard several times through the conference, is the enhanced permeability and retention effect, or EPR effect. And we see that for particles that are anywhere from about 20 nanometers clear up to several hundred. We put the upper limit of our interest at about 220 for 2 reasons. One is that if particles are greater than that, greater than about 230, 240, they'll get caught up in the architecture of the spleen and liver. And also 220 is a common filter size for a lot of biologics. Okay, so what do we mean by size? Well, what I'm showing you here is size of a particle monitored by 3 orthogonal techniques. The first being electron microscopy, then atomic force microscopy, and also by dynamic light scattering. You can see that these are commercially available gold particles. You can see that their numbers correspond very nicely. For the naked particles, particles that have been unexposed or unmodified, we get pretty close to the manufacturer's specification of about 30 nanometers. But what happens if I incubate those with a physiologically relevant buffer, or more specifically in serum, plasma? What we see is that by TEM we do not monitor a difference. By AFM we do not monitor a difference. But look what happens by DLS. The particles more than double in size. Again, this speaks to the measure and that we're looking at when we monitor things. So what you're seeing there by DLS, as was discussed earlier, is the hydrodynamic size, the tumbling effect of those particles. So due to the opsins, the proteins that are coated onto the surface of the gold, when it's incubated with plasma, that size grows. And I just showed you in the previous slide why that is important. So it's very germane to characterization to understand how that particle behaves in an in vivo system, because it's not the same size as under pristine laboratory conditions. We often see images like this of the very best image that they've captured in the last 3 years of their particle, and look how homogeneous it is. There's no problem with this whatsoever. But then upon closer inspection, we often find that that's not quite the case. And so how do you monitor something like this? How do you call the size of this polydispersity? Well, you tell me. We just provide the data. So by TEM, we get a size of roughly 14 nanometers, and we literally just count particles, several hundred particles, to come up with that histogram. By DLS, though, because of this tumbling effect, the hydrodynamic size is more on the order of about 80. So again, how do you call that size? You tell me. We just report them. Okay, so why is this important? So I'm going to give you an example of a case study of how a difference in size, and specifically a difference in surface chemistry, definitely contributes to biocompatibility. You're not going to be able to read this other than to show you that we received 2 batches of material, and were able to characterize them under various means. What really got our attention, though, is the first batch showed fairly severe toxicity. We saw lesions in the lung, and several animals died. But yet when we reported that back to the sponsor, my standing joke is the CEO says, hey, NCL, those particles were working great when we gave them to you. What did you guys do with them? No, we just call them like we see them. So we were, through coordination with that company, we were able to get a second batch of material, and interestingly enough, that second batch was fairly benign. We did not see the lesions. But by all of our PCC methods, DLS, EM, we were not able to detect a difference. Normally that's a tip-off. If we identify some difference in PCC, we're able to say, okay, this is likely the parameter that influences that toxicity. But again, we saw nothing by PCC. So then we introduced the particles to a derivative of what I showed you before. Is we incubated the particles in serum and looked at the difference between the 2 batches as far as which proteins absorbed on the surface. And although you can barely see it here, there was quite a difference between batch 1 and batch 2, where batch 1 had far more proteins absorbed on the surface, which led us to believe that there was something happening with the PEG layer. We developed an assay to look at that. And I apologize, it's very probably very small here, or at least it is on my screen. You can see there, if you look at batch 1 versus batch 2, as far as the supernatant, which would be the free PEG in solution, the PEG that has come off of the particle, there's far more PEG in the supernatant of batch 1 than there is batch 2, whereas for batch 2, the majority of the PEG is still on the surface. The company was then able to use this as a QA/QC method for lot release criteria. Here's another example of more or less the same thing, and it shows the in vitro, in vivo correlation that we're able to see on some specific assays that we have. Here's 3 examples of particles that are either unpegylated or pegylated, especially these 2 here. What we see for the phagocytosis is, if you can see these black spots, the electron-dense regions are due to the gold that's phagocytosed by the macrophage, whereas the pegylated version has hardly any at all. That also correlated to an in vivo response where the gold was deposited both in the liver and the spleen. I should say that even though these are black due to the amount of gold that's present in there, it turned out that gold is very benign. The company then later optimized it and pegylated it, and here's a result from a subsequent batch. Then I showed you this example here. So indeed, surface modification is critical, and the stability of that surface is very essential to biocompatibility. Just 1 or 2 other examples of in vitro to in vivo correlation. The beauty of nanotech and nanomedicine is we're able to reformulate drugs that are extremely toxic, and rather than getting a full systemic exposure, hopefully the amount in the tumor due to EPR effect increases in many cases by orders of magnitude. Here's an example of a hemolytic API, hemolytic drug that was reformulated into a micelle, and by PCC it looked pretty good, but what we found is some of the drug was missing. If you can see it here, the theoretical versus the measured, we're missing roughly 75% of the API, so where could it be? Well, we suspected that it was just free in solution. We saw that in vitro, where the formulation was very hemolytic, meaning it lysed the red blood cells, and that mapped also with in vivo, that we saw very severe hemolysis of the red blood cells. Ideally, I should back up that we monitor a whole set of parameters in our characterization assay in PCC through in vivo, and it's very difficult to get everything just right because in addition to the PCC, you have to have a nanoparticle that releases the drug on kinetics that's consistent with efficacy. So if it's too stable, it will not release, and if it's unstable, it will release in a burst fashion within seconds to minutes. So in addition to that, just monitoring those various species is a challenge in itself, and we have a number of assays to do that. I should mention again, and I said this on Monday, there's many fractions to a nanoparticle formulated drug. There's the there's the encapsulated fraction, there's the unencapsulated fraction, there's the protein-bound fraction, and there's the free fraction. Whereas it's important to know that the toxicant and also the drug that is most efficacious is that unbound free drug. And so as we monitor it, we have to make sure that that is the right component of the total fraction that we're looking at. So in the interest of time, I'll go through this very quickly, other than we are seeing, again, in vitro to in vivo correspondence for things such as drug release. And again, it can be very complicated if you have something like a prodrug that is released and then cleaved. it can be difficult to mimic that under in vitro conditions, although we've had quite a bit of success doing so. One of the active areas of basic research in my laboratory is Marina Dobrovolskaya's work in looking at nanoparticles' interaction with the immune system, because as I mentioned, and many of you know, the immune system is likely going to be the first biological barrier that they come across as far as clearance of the material rather than excretion. It's going to be likely phagocytized. So optimizing that can make or break a particle, especially when efficacy is involved. Here's an example of something that we're actively involved in, and that is the release of cytokines and complement activation. In cancer trials, it's not required until phase 2, but why not do that early on if we have an in vitro assay that maps very nicely to those in vivo responses? Marina has published quite a bit of work on this, and we strongly encourage you to take a look at complement activation as you evaluate and do preclinical characterization. We'll also say that because of the genesis of nanomaterials, where often the materials came from engineering labs or material sciences labs or chemistry, where those laboratories may not be very familiar with aseptic techniques, roughly a third of the particles that come into NCL we have to send back because they're contaminated with endotoxin. Now, endotoxin is a very powerful ligand for the immune system. We get charged up, our immune system gets charged up if we think that there's a bacterial contamination, and endotoxin is a derivative of the bacterial cell wall. So just as a word of caution, make sure that you use pyrogen-free glassware and go to great lengths to avoid endotoxin contamination. So in conclusion, let me recognize, again, the true brains. I'm very proud of this team. I'm on the road a lot, but these are the real smart guys. So Anil Patri is my deputy, and he's in charge of the chemistry section. Steve Stern is our toxicologist and runs the majority of our in vivo studies. And Marina Dobrovolskaya is our immunologist and has published quite a bit of work on the interaction with nanoparticles in the immune system. And of course, Jennifer Grossman is our main conduit for most of our sponsors. Okay, thanks for your time, and I guess I have questions at the end.
Thank you very much. Any burning questions? Please go ahead.
You mentioned the hydrophobicity is one of the important property, physical chemistry property of nanoparticles. How did you measure hydrophobicity?
Yeah, that's a great question. And if anyone has an assay to come up with that, we've been asking for one for 10 years. So indirectly, it amounts to the degree of pegylation. So we cannot get a true number on a score, on a scale of hydrophobicity. It's not like the partition coefficient. So we, as I said, indirectly will monitor pegylation, both the density of that peg as well as the length of the peg.
Oh, yeah. Thank you.
Yeah, yeah. So the comment from front was through contact angle, we can monitor the density of peg, and yes, we're actually getting into that now.
Also hydrophobicity. It's the classical, always working method is contact angle.
Mm-hmm. Okay.
What is your latest opinion on release test media being biorelevant for different species, or in general, what kind of criteria would you put to make it biorelevant?
For release of the drug from the nanoparticle?
Mm-hmm.
So we only use rodent models, and specifically for PK studies we do rodents. And as you just identified, there's complications to that. Presently that's the best, cheapest method that we have. We also will do ex vivo release that appears to correlate fairly nicely. Yeah. But as far as which model to use for which clinical indication and for immunotox, do you have to go immediately into a non-human primate model? That's a decision that we have with the company because If, for example, they have a protein-based nanoparticle, immediately what comes to mind is immunotoxicity, that you're going to get an antibody response. Yet you will not see that in a rodent model. It's just not relevant. So then to get at the true toxicity often requires far more elaborate techniques than we can offer them.
Okay, thank you, Will.
Oh, okay.
Oh, I missed you. Okay, sorry.
Is this service open to other countries outside the U.S.?
Oh, absolutely, yes.
And are you just restricted to cancer alone or other diseases?
Right. So your first question is, are we available to international? Absolutely. As long as there's a non-binding agreement that that company will eventually submit their application to the FDA. Yes, we can. We're open to cancer is what I can offer for free. If there's other clinical indications, then I have to charge for that. Okay, thank you.
Okay.